we're going over 8.4, which is fairly short, and then we're going over the first half of 8.5 because they're two processes, very important processes that happen in our cells, and they go together, transcription and translation. Sorry. And then next week, we're going to go over the next half of 8.5 and 8.6 because those two go better together. So this is just how we group them. So our central question for 8.4 is what happens during the process of transcription? Transcription converts a double-stranded DNA molecule into a single-stranded RNA molecule. So in 8.2 and 8.3, we talked a lot about DNA, how it's double-stranded, how it gets replicated into another DNA molecule. Transcription is actually a very similar process. But instead of getting another double-stranded DNA molecule, we get an RNA molecule. So the central dogma. A dogma is just a set of beliefs. This is a central statement to the field of biology. So the central dogma of biology states that information flows in one direction from DNA to RNA to proteins. And that's just illustrated in the picture. DNA copies itself, DNA can be turned into RNA, and then RNA gets turned into proteins. The other important part of this picture is the location. We'll add to this picture on the next slide in a minute. The processes that happen during the central dogma. Replication we talked about last week. And then transcription and translation are the other central processes. We will talk about that today. So we had DNA copying itself. That is the process of replication. That's what 8.3 was all about last week. When DNA gets made into RNA, it is called transcription. Both of these processes happen in the nucleus because that is where the DNA is found. And then when RNA gets turned into proteins, that process is called translation. And that process happens in the cytoplasm. RNA is the middleman between DNA and proteins. DNA is huge. It is 3 billion base pairs long. It doesn't fit outside of the nucleus, so it has to send a messenger. So that middleman messenger is the RNA molecule. And yes, it says draw this picture, so I would like you to draw this picture in your notes. I don't have you draw a lot of things, but there are things that I do want you to draw, and this is one of them. And make sure in your picture you have the locations, what happens in the nucleus and what happens in the cytoplasm. That is an important part of this picture. So a little bit about the structure of DNA versus RNA. So all the DNA stuff we should already know, should be review. We went over it the last 2 weeks. DNA, part of it is made of a sugar called deoxyribose. That's what the D in DNA stands for. You have the 4 nitrogen bases, A, C, G, and T. We should also know by now what goes with what. A and T go together, C and G go together. And it is a double-stranded molecule. It has that double helix, the 2 strands twisted around each other. And then we compare it to RNA. The R in RNA stands for the sugar ribose. It also has 4 bases, but its 4th base is uracil instead of the thymine, the T. So A, G, C, and U stands for uracil are the 4 nitrogen bases. And it only has 1 strand. When it gets made, it only copies half of the DNA. And we'll see how that happens here in a minute. So there are 3 steps to transcription, just like there were 3 steps to replication. And the first step is the same. The DNA strand has to get unzipped. So step 1 is initiation. An enzyme, helicase, comes through and unzips. I don't know why this picture is here. This is the picture for the next one. I'll fix that later. But helicase comes and unzips a segment of DNA at the start site. Step 2 is called elongation. And that word in general just means to make something longer. But free bases come in and pair with 1 strand of the DNA, not both. The C's and G's, the curly letters, they still stick together. And the DNA strand will still have a T, so it will attract the A's. But the A's from the DNA strand will now attract the U's for uracil because there are no more T's. And it is directed by RNA polymerase. That is the name of the enzyme. That completes this part. So you can see in the picture, in the GIF at the bottom, DNA strand gets unzipped, comes in pairs with 1 strand, and the A's attract U's. And then step 3 is termination. Termination means to terminate, stop. So enzymes like ligase bond the nucleotides in the mRNA strand together to make a single strand of bases. The mRNA strand separates and moves out of the nucleus into the cytoplasm. And it is looking for a ribosome to complete the next part of the process. And the DNA strand just rewinds and stays in the nucleus. So this is the end, the termination of transcription. That red strand being made is the RNA. It reaches it's called a terminator sequence that tells transcription to stop. And the RNA strand just floats away. It is small enough to move outside of the nucleus and go to the next part. So we are moving on to translation. Like I said, we'll do 8.4 as transcription. It's fairly short. And then we're going to move on to translation, which is the first half of 8.5. So our essential question, what are the steps in the process of translation? And they are very similar. All these steps to all 3 processes are similar. So in general, the general definition of translation, it converts the message encoded in mRNA, the mRNA strand we just made, into a protein. And then in the picture, in the animation, you can see all of the processes happening. You have DNA, gets made into RNA, moves out of the nucleus into the cytoplasm, and then a protein gets made in the end. Let's see. Overall animation of all three of these processes. So we have to go over some vocabulary when we talk about translation. So the next 2 or 3 slides are general vocabulary, and then we'll put them into the processes. But codons are nitrogen bases grouped into 3s on the mRNA strand. And each codon gets translated into a different amino acid, which, as we talked about way back in chapter 2, are the building blocks of proteins. So in our example at the bottom, our codon is AGU. We always talk about these things in sets of 3. And codons will attract its mates. The A, the G, and the U on the codon will attract its mates on something called an anticodon. So the codon on the mRNA strand attracts its complementary bases, just like we've been talking about for the past 2 weeks. The complementary bases are called anticodons, and they are found on a molecule called tRNA, or transfer RNA. And yes, this is another picture I want you to draw. It doesn't have to be perfect. It doesn't even have to be in shapes. The point is, the sets of 3 on an mRNA strand are called a codon. The sets of 3 on a tRNA strand are called anticodons. And they join together. They are attracted to each other. Well, welcome to class if you just got here. Let's see. Cashley, good morning, Cashley. Good morning, Daniel. Good morning, Jackson. Good morning, Maya. Maya, you're new. I'll talk to you after. That's okay, Daniel. You can watch the video this afternoon when we're done to get it all. So tRNA is called transfer RNA. That's what the T stands for. So each tRNA carries the amino acid on top and then the anticodon on the bottom. So to me, this looks like a little bent over old man. He has 3 little feet. That's his anticodon. And then his face is the amino acid on the top. So mRNA is a strand. tRNA is a different looking molecule. We're going to, like I said, put all these vocabulary words together so we know what's going on during the process of translation. So this is review. We're already done this, but at the end of transcription, the mRNA strand comes to the end, the terminator sequence. And then it detaches and then leaves the ribosome, or leaves the nucleus, to go into the cytoplasm to find a ribosome to attach to. So step 1 of translation is called initiation. To initiate means to start. And then in the picture, it happens quickly, but the mRNA binds to the small part of the ribosome. So that came in first. A special initiator tRNA binds to the start codon on the mRNA. AUG is always the start codon. Those 3 letters signal the start of translation. So remember, this is the mRNA. So we had AUG. That's the start codon. This is the tRNA. It looks a little different, but you got the anticodon on the bottom and then the amino acid on the top. And then the large part of the ribosome binds to the small one on the bottom. So I think if I want to see that again one more time, I can. There we go. So the small part comes, then the tRNA, anticodon, the codon come together, and then the large part binds. And now we have, it's called the initiation complex. We have all these 3 molecules together ready to start translation. Step 2 again is called elongation because it just gets longer. So the mRNA attracts its complementary bases on the tRNA. So the codons attract the anticodons in sets of 3. We always do it in sets of 3. And then the amino acids on top bond to each other. And then in the end, you'll see it'll be a long chain of amino acids, which is what a protein is made of. So we have all these codons are going to attract the anticodons. And then these amino acids at the top are just going to start binding to each other. Again, if I start it over, it bonds to each other and then it moves over. There's the next one. And then it moves over and then another one's going to come in. And this process happens over and over again until termination. So elongation continues until a stop codon reaches a certain spot on the ribosome. The completed polypeptide, which is another fancy word to say protein, is freed. And then the ribosome splits back into smaller pieces.